moving on to our next keynote speaker, Mr. Frank Orden is president of the Julius Kühn Institute Federal Research Center for Cultivated Plants in Germany, an honorary professor for molecular resistance breeding at the Martin Luther University in Halle-Wittenberg. His research focus is on molecular plant breeding with special emphasis on improving resistance to biotic and abiotic stress. While the Julius Kühn Institute deals with a much broader research comprising of plant protection and plant health as well as agroecology. Frank will present his scientific paper on resistance breeding, the cornerstone for meeting future challenges. Please welcome Frank.
Yeah, Johanna, thanks a lot. Good morning, ladies and gentlemen. Monica already mentioned plant breeding, and I will give you a little bit more detail now. As we all know, our planet is under pressure, as in 2050 we have to feed 10 billion people, and that's in the background of climate change, of preserving biodiversity, and of crisis. Actually, the Ukrainian war having strong impact on food supply. And in this respect, efficient plant production is of prime importance. When we look at the plant production chain, yield is the result of the yieldability of the genotype, the cultivar, and its interaction with the environment, means soil, weather, and biotic stress, as well as with management practices like plant protection, fertilization, and as we heard several times today, the farm-to-fork strategy recommends a reduction of 50% in pesticides, 20% in nutrient application, and so plant production will lose management options. And in this respect, it is of prime importance that yieldability of the genotypes will be improved and its interaction with the environment, on the one hand, with abiotic stress, and on the other hand, in the background of losing pesticides with respect to biotic stress. And this is of prime importance, and this was mentioned already today also. For example, in wheat, the use of pesticides saves a 90% yield, but much more in potato due to Phytophthora infestans. So, let's say, plant breeding has very major challenges to contribute to a more environmental friendly agriculture. And this all has to be done in the background of climate change. For example, a rise in temperature on 2 degrees leads to 230 million tons of loss in wheat, maize, and rice due to insects. And of course, when climate gets warmer, insects will move north. And the same holds true for fungal pathogens, which are adapted to more warmer temperatures here in Europe, like stem rust in wheat, and of course, insect-transmitted viruses like turnip yellow and barley yellow dwarf virus. So there is really a large task for plant breeding. And if we look at this, plant breeding in the past has been very successful already. I won't go into detail with this very busy slide here, just to show you wheat cultivars released in the 1960s till today were tested on multi-location, multi-field trials, and you see the younger cultivars here in yellow, and as you see here for grain yield, for protein yield, for nutrient use efficiency, the younger cultivars are outperforming the older one. And if we now look in more detail with respect to resistance, we can see here that during the time, the susceptibility to stripe rust and to powdery mildew was considerably reduced. And this independently from the application of nitrogen and of pesticides. And just to show you, here you see an old cultivar called Topfit, released in 1972, completely covered with powdery mildew. The same trial in newer cultivar, you don't see any symptoms of powdery mildew. So, this success in plant breeding was mainly achieved by conventional pedigree selection. This means phenotypic selection was conducted during segregating populations. Breeding of new cultivars, you can also see, is quite a long task, taking about 10 years. This can be abridged today and is done in wheat and barley routinely by the use of double haploids, which are homozygous in the F1 already. And so this leads to a speed up in plant breeding, and since the 1990s we can use marker-based selection. For major genes, you see here the gene SM1, and this confers resistance to the wheat orange blossom midge, a trait which you cannot select by phenotypic selection. And as Monica already mentioned, Cymoseptoria tritici, one of the most important wheat diseases today, you can also use these markers to identify genomic regions which contribute to this resistance, means which explain a certain amount of the genetic variants you see, and so you can introduce these resistances by molecular markers. So, especially in the last decade, very efficient tools were added to the plant breeder's toolbox. And this is especially with high throughput marker techniques. Today, you can screen hundreds, thousands of loci within a genome in a very short period of time. And we also have in most of our crop species the whole genome sequence available. For example, in wheat and barley, in potato, or in sugar beet. And this has a great impact on plant breeding and the time you need to breed new cultivars. I will come to this later on. For example, combining these high throughput genotyping systems with phenotyping systems today allows the systematic use of plant genetic resources which are stored in many countries and in Svalbard as well. And this is a key to solving future problems. And we can do this by applying genome-wide association studies, a technique which normally comes from human genetics, correlating the genotypic data to the phenotypic data. And here you see an example. At the IPK in Gatersleben, which is the German gene bank, 20,000 barley accessions were tested analyzed with high throughput marker systems, and around 3,000 of these were tested for resistance to barley mite, barley yellow mosaic virus. And what you can see here, you can see significant associations here on chromosome 3. That's a gene which is effective against barley mite, barley yellow mosaic virus, and here another one effective against barley mite mosaic virus. So without doing any crosses or segregating population, which takes time, you can develop markers for these genes and use these directly in breeding. Furthermore, in plant breeding now, genomic selection can be applied, a procedure which is applied in cattle breeding for a long time already, but this requires a lot of molecular markers. And it's that way you have a training population, this is genotyped and phenotyped, a model is trained, and in your breeding population, then you only do genotyping, calculate a genomic breeding value, and do selection on this. And the big advantage of this, you do not do phenotyping anymore. You just propagate, calculate these genomic breeding values, and as you can see here, for Fusarium head blight in wheat, the correlation between the observed and the predicted value is around 0.2. Therefore, genomic selection can be applied in plant breeding or in resistance breeding today. And I mentioned already, we have these high throughput marker systems, and we have the sequence of our genes. And in former times, to identify resistance genes, this was a very time-consuming progress, because you have to do marker saturation, you have to construct bug contigs, which then have to be sequenced. Today, with our high throughput marker system, this is very easy, and then you can identify candidate genes because you know the sequence. And why is this important? Because when you combine genes with a different mode of action, this may result in longer lasting resistances. And this is shown here. Long time ago, we isolated the resistant gene RYM4, effective against barley mite and barley yellow mosaic virus. And it turned out that this locus encodes a translation initiation factor. And what does this resistant gene do? It interacts with the viral genome-linked protein of the virus, and if this here fits, then translation starts and the cultivar is susceptible. We identified another resistant gene, RYM11, against this disease, and that's a protein-disulfate isomerase. What do these genes do? They fold proteins correctly, and if proteins are not correctly folded, they are ubiquinated and degraded. So, and the resistant gene is a loss of function allele. And so when you combine these resistant genes, so-called pyramiding, this may result in longer-lasting resistances, this gene interacting at the very beginning of the virus replication, this at the very end in protein folding. And also knowledge on resistance gene is a prerequisite for allele mining. Now you can go to these large gene bank collections, just sequence your gene and identify new, maybe more powerful alleles. This was done here again on the sequence for RYM11, and you can see here 4 different alleles were detected. However, these genome sequences, or these resistance gene sequences, are also a prerequisite for genome editing. And genome editing is a very powerful tool which was added to the plant breeder's toolbox. I cannot go into details, but all in all, by different systems, cutting enzymes are led to a predefined location within the genome, and then these enzymes lead to a double-strand break. This double-strand break is repaired by non-homologous end joining. And in this process, mistakes do appear, leading to insertion or deletion of base pairs or to gene disruption. And on the other hand, you can also use this by homologous repair. You give a homologous template with a mutation, for example, which is then incorporated. And of course, it can also be used to incorporate longer sequences. That's gene technology, and new techniques even go to the exchange of single base pairs. So just we stay with the barley mite, barley yellow mosaic virus resistant RYM4, the translation initiation factor. In collaboration with the IPK, we used CRISPR-Cas to induce these mutations here, and when these plants are tested for resistance to barley mite mosaic virus, they were all resistant. And what is most important, you can use a high-yielding cultivar, which is very fine with yield, with quality, and only one resistance is missing. If you know the gene, you leave all the nice traits combined and just add these genes. For example, when we look here, you see these are the CRISPR-Cas treated ones. This is the high-yielding variety. You see no difference in 1,000 kernel weight. Why here you can see a little bit of reduction in the number of grains per plant. But all in all, this saves very much time when you edit this resistance by CRISPR-Cas. And this is done all over the world except in Europe. Because in 2018, the Court of Justice said that organisms generated by target mutagenesis underlie the strict regulations and are not exempted from any obligations. This means also this here, SDN1, this gene disruption, the result of this CRISPR-Cas treatment, you cannot distinguish from a natural mutation, but nevertheless, it is gene technology. That this here is gene technology for sure when you introduce foreign genes. So, coming to an end, I hope I could show you that improvements in plant breeders' toolbox, for example, the availability of high-throughput marker technologies, genome sequences, and so on, and the possibilities of genome editing have transferred plant breeding to a new era. Knowledge on resistant gene sequences will facilitate efficient allele mining as well as a targeted editing of respective alleles, for example by CRISPR-Cas, and resulting in a much faster combination of resistance with superior agronomic performance. So, modern breeding for resistance will be the cornerstone for an environmental friendly and productive plant production in the background of climate change and the loss of insecticides and fungicides, thereby contributing to the aims of the Farm to Fork strategy. Thank you very much for your attention.
Thank you so much, Franck, for this extremely explicitive and quick, however fascinating presentation on breeding. Who would like to ask a question to Franck? Over here on the left.
Thank you very much for this nice talk. When we talk about breeding strategies, you evoke all of the end targets: ecological services, yield, resistance to pests. What about impact on human health, nutrition? And especially when we talk about wheat as one of the main guilty partners in obesity and diabetes and everything.
Absolutely right. Let's say a new cultivar is the combination of all traits. And for example, in wheat, of course, people are working for long. One quality parameter of wheat, of course, is baking quality. No? This has been improved very much. Let's say today you can have the same loaf volume with a lower protein content, but what is an ongoing research objective is to reduce the allergy of gluten. And this is still ongoing, and of course it's also a very important trait. And research on this is done all over the world.
Okay, does that answer your question?
Yes, I mean, these things can be incompatible. The two end goals can sometimes be.
No, let's say this is not very likely in that case. The problem is that gluten is the protein which really makes the extraordinary quality of wheat. It's making these loaves, no? And so to me, there is no disease resistance known which, for example, is related to the gluten content or the composition of the gluten.
Okay, but that's one example. But if you look at resistance to insects, insects are often attracted to the same foods that are high in the proteins that we need because we have the same needs in amino acids.
Yeah.
And so some of these things, questions, I just think it's important that we need to think about both things. We're producing food, but why are we producing food? We're producing food to feed people, to maintain health. And sometimes those questions are, I think, not well enough integrated into the discussions that we have.
But you're absolutely right. For example, lupins. Now, we are growing lupins, and lupins are, let's say, very much affected by aphids. No? And let's say, especially the sweet lupins. But what we found out that there is in some cultivars, there's a special alkaloid in a slightly higher concentration, and this leads in a higher resistance to aphids. No? But let's say they are still sweet. No? And so there is an interconnection which we have to have in mind. You're absolutely right.
Okay, thank you. Other questions, please? Yes, Christian, and then we have a question also on the stage.
Thank you, Franck, for your presentation. It has been briefly introduced by Ricardo that we have to focus also on the microbiome, and the plant has a microbiome on the leaf, on the root, even an endosphere. What do you think about moving from the concept of plant breeding to a breeding for the holobionts?
Christian, that's a good question. Not we are working on this, but what has been shown that certain genotypes generate their own microbiome, no? And that the microbiome has an impact on, for example, resistance. And let's say this is just basic research, but it's research on optimizing this interaction, microbiome genotype. No? Let's say to make plants which are more resistant or tolerant to biotic and abiotic stress. That's a very interesting research area which has just started, and we are really in the basic research area there.
We have a question on the stage. Can we also... okay, while we're getting this mic taken care of, there is a question also in the middle. Okay, we'll answer the question on the stage and then we'll go back to the public. There we go. No problem.
Yes, I have a sort of a follow-up question on that question, because the CRISPR-Cas is something very specific, you know, which gene is doing something, and you have a limited number of genes. How much should breeding spend on that kind of research, of that kind of breeding, and how much should it spend on increasing the overall stress tolerance of the plant? So not specific resistance traits, but overall stress tolerance.
Yeah, that's a good point. For example, these genes, these major genes I've shown you, they are overcome at certain times. No? But let's say when you do this, for example, the Simosatori artritici, that's a QTL. It does not lead to complete resistance. No? So let's say so there will be no selection on the pathogen, and they will be longer lasting. No? So, but let's say the problem is plants need to have a certain level of tolerance against abiotic stress and against biotic stress. But for some pathogens, as Monica has shown, there is really breeding for resistance is needed. And in case we lose management option, and I did not know that in Denmark even copper is not allowed anymore up to now, then let's say we really need resistance in these cultivars, because
we should decrease it, but should we spend more energy on the stress tolerance resistance breeding?
Do you mean abiotic stress or in?
Biotic, a little bit like Christian was saying on the biomes and this kind of.
Yeah, let's say it's just started. No? To take into account much more data. No? For example, data on climate, on soil water content, all this, let's say at the moment also artificial intelligence, combining all these data just in the basic stage, turns into plant breeding. And so let's say, in my opinion, we need a high level of tolerance to biotic and abiotic stress, and in addition, we need resistances based on certain genes or QTLs. This we need, of course.
Okay, and one last question from the audience, please.
Yes, thanks, Frank, for the great overview. My question follows up on the discussion that Christian initiated to expand the view of breeding to the microbiome. I would even expand that further to the cropping system and the landscape level. And my question refers to one of your first slides where you crossed out the M. I think is not in the systems we are discussing and the diversified toolbox that we've mentioned before. The M, the G times M, particularly important to understand and consider in the breeding activities.
Absolutely right, Frank. When we have new breeding systems, like new cropping systems, no? For example, let's say the usabilities for crop mixtures will be a breeding goal. No? If we lose more herbicides, let's say ground coverage in the youth development, no? Will be a breeding goal. And so let's say we have to take all this into account, and in the end, but that's difficult. We should end up with cultivars which are best suited to the location and management practice they are used for. But that's a long-lasting and difficult way. And in this respect, also all this information on weather, on soil water content, on all these things is needed to breed these kind of cultivars.
Okay, thank you very much. That's all the time we have for questions.